Hey, welcome everyone, and thank you for joining us for today's webinar. I'm Steve Daniel, Chief Business Officer of Empiri. I'm delighted to host today's session titled Empiri eSlice, a TME preserving ex vivo platform for cancer agnostic efficacy, target validation, and MOA evaluation. It's my privilege to introduce today's speaker, Dr. Kyu-Sun Yoon. Dr. Yoon is an associate professor of neurology at Houston Methodist and Cornell Medical College. She's the inaugural Houston Methodist Chair of Neurodegenerative Disease Research and is co-founder of Empiri, a precision oncology biotech company and the sponsor of today's webinar. Dr. Yoon earned her BS and PhD in biology from Caltech, completed postdoctoral training at UCSF and Dartmouth, and held a faculty position at the Jackson Laboratory before joining Houston Methodist. Dr. Yoon's research evolved from neural stem cells and brain development to cancer stem cells and brain tumors, and is now focusing on tumor-immune interactions. At present, Dr. Yoon's lab explores sex-biased immune roles in brain cancer and neurodegeneration, molecular and cellular heterogeneity in diseased and healthy brains, cell-cell interactions in glioblastoma, medulloblastoma, Alzheimer's, Charcot-Marie-Tooth, and other neurodegenerative diseases. There will be an opportunity for question and answer after the presentation, so feel free to submit any questions in the chat window to the right. And with that as background, I'll turn over the podium to Dr. Yoon.
Thank you, Steve, for that very comprehensive background introduction. I wasn't expecting that. And welcome, everybody. Thank you for joining us this morning. I'm delighted to share the next hour with you or so and tell you about a novel 3D culture method that I have developed and Empiri's is licensed to use. And also, I would like to make it clear that this technology we have opened up to all researchers in academia and industry. And so to enhance and accelerate your drug discovery or research projects. So please feel free to think about how you can use this platform. And if you have any specific questions, put them in the chat or we can discuss it at the end of the presentation. So I'm going to try to keep it short so we can have a lot of good discussions afterwards. Oh, first technical challenge. Why isn't this working? Oh, there we go. So I mean, from the fact that you signed up for this webinar, I assume that you are very familiar with the importance of the tumor microenvironment and the failure of the cell lines that we've been used in the pharma for the last 100 years to develop anti-cancer agents. And I think this a slide summarizes some of the advancements we've made going beyond the established cell lines to model human cancer ex vivo, and also, but there are a lot of limitations to these models as well. So for example, PDX models where you take patient tumors and xenograft them into immune deficient mice obviously lacked tumor stromal microenvironments, especially the immune cells. And more commonly used or now becoming much more popular models such as organoids and PDCs, again, suffer from similar kind of challenges in that they don't maintain the tumor microenvironment that's unique to each patient at different stages of tumor progression. So, understanding that human cancer is a very dynamic ecosystem composed of multiple cell types that communicate with each other, there really is a desperate need to develop an ex vivo system that's amenable to experimental manipulations, relatively low in cost and time it takes to generate them, and faithfully model the human cancer as closely as possible. So that's why I developed this method called eSlices. And a major differentiator between eSlice and let's say organoids, which are also 3D culture methods of human tumor, is that organoid systems mostly start with its first step is to dissociate the tissue and generate this single cell suspension that then it grows out ex vivo, often in the presence of artificial stimulants or serum that can change the cellular behavior, especially immune cells and cancer stem cells. And there's a significant amount of culture selection as you expand these ex vivo. In contrast, eSlices are generated from fresh patient tumors and immediately exposed to treatments. And importantly, it maintains the tissue architecture because there's never a dissociation or reconstitution of any type. And so we can maintain the important cell-cell interactions, cellular juxtaposition, as well as extracellular matrix. And as I show you at the single cell resolution, maintenance of tumor-educated and associated immune cells and stromal cells. So, I mean, just to highlight the importance of the tumor microenvironment, I just want to just show you this one anecdote or short story from a work that we published from my academic lab, where we did massive single-cell sequencing from human glioma patients. And one of the goals of the study was to understand the importance of the tumor-associated myeloid cells. So we sequenced over 87,000 human glioma-associated myeloid cells and identified 9 different molecular subtypes of myeloid cells. And on the right, Kaplan-Meier curves show that when we take the gene signature for different myeloid cell subtypes, while ignoring everything else about the cancer, the mutational profile, the molecular phenotype, etc., we can actually stratify patient survival, meaning we can predict how well or how poorly the patient will do depending on the kinds of myeloid cells that are present in that particular patient. And so I think this example really underscores the importance of having those myeloid and T cells and fibroblasts that are in the tumor microenvironment that are non-cancer, but as important in terms of determining disease progression and therapy response. So what is an E-slice? E-slice is a precision-cut live tumor tissues cultured in chemically defined medium. So this is our proprietary medium, and is compatible with either needle biopsy cores as small as 18-gauge needle cores that will be compatible with prospective clinical trial studies, as well as large tissues that we can get from surgical samples. And so the assay is scalable depending on the goal of the study, from either clinical trial all the way to research and drug discovery and development. And it maintains the native tumor microenvironment and cellular heterogeneity, including immune cells and cell-cell interactions. A key to that differentiates our platform from other tumor slices or organoid slices or tumor fragment studies is that we have a proprietary method for measuring tissue viability without destruction or fixing the tissue that allows us to do longitudinal monitoring of dynamic treatment responses. What that means is that we built in multiple internal control quality control steps that allows us to account for spatial heterogeneity of cellular composition, excluding regions that has mostly necrotic tissues, etc., before we set up our treatment groups, and so we can do fair comparison across the samples from the same patient. It's amenable to chemical, physical, and biological manipulations. It means we can do, testing of chemotherapies, targeted therapies, oncolytic viruses, biologics, cell-based therapies, etc., and perform multiple perturbations from the same patient tumor on the same day. So it's about as closely controlled experiments as you can perform with a human tumor. And this can be used to test drug dosing, drug combination, or scheduling of different drugs. And we do see significant effect with the different scheduling. So obviously it's cheaper and faster than PDX models or GEM models, and allows, importantly, monitoring of immunotherapy responses ex vivo. And that's, again, coming from tissue-resident immune cells as opposed to artificially added PBMCs or other systems. And we can also perform multimodal sample analysis at endpoint or during the different time points in the analysis to gain molecular insights, discover biomarkers, and test different combination therapies. And importantly, I will show you 2 retrospective clinical studies and 1 prospective clinical study that's ongoing that show that this assay can accurately predict cancer patient responses to treatments. But there are limitations. We are not hooked up to the circulation, so there are no new immune cells coming in or out of the system or going into the lymphoid organs. And so our ability to measure immunotherapy response is restricted to what's in the tumor tissue. However, we can add exogenous factors or cells depending on the experimental goal. And of course, the pharmacokinetics can be very different ex vivo in people. So basically, Empiri's workflow is we either take a biopsy or surgical material, and for our clients, we actually procure the samples for you. And many of these are often shipped overnight on ice. And so if you're doing clinical trials outside of Houston, Texas, no problem. You can just ship us the samples. Or if you have samples from mouse models or PDX models or your patients that you like us to perform e-slice assays, again, you can just ship it on ice overnight without any compromise in viability. And we make these precision cut slices, and then we expose them to drugs immediately after the initial quality control steps. And then because we have baseline viability readings from every well, we can measure both absolute response from each well after perturbation, either drug addition or control vehicles, as well as measure relative response on a fixed day, let's say day 8, compared to vehicle control, how these different drug combinations may have affected viability. And then we can generate a report. So in the clinical diagnostic setting, which is the original application that I had in mind for this platform, we can provide a report in 8 days. And just those of you who are curious, we are a CLIA registered laboratory. E-Slice has been tested in all major solid tumor types and in treatment modalities, alone and in combination. These include GBMs, medulloblastomas, because of my academic interest, as well as head and neck cancer, breast cancer, lung cancer, liver cancer, ovarian cancer, stomach cancer, colorectal cancer, bladder, and pancreas. And as you can see on the pie chart on the right-hand side, most of our assay development, as well as validation and ongoing studies, are with human tissues, either from PDX models or from clinical samples directly. And as you can see, about 30% of the samples are from patients' tumors. And on the pie chart on the right bottom, you can also see quantification of different tumor types we have tested thus far. And for the researchers in the crowd or in pharmas, you would like to do a deeper analysis of how your agent may affect different cell population or beyond just the viability change. Again, this was developed from the beginning to be a clinical diagnostic assay. We wanted to mirror the clinical monitoring of patient tumors as close as possible. So that's why we measure viability from the entire tissue. However, we can collect and analyze, for example, conditioned media, which is chemically defined. So it's a beautiful platform for doing biomarker analysis for secreted proteins, metabolites. We can take the tissue at any time point and fix them or lyse them for RNA and protein analysis. But importantly, the real power is coupled when you couple the e-slice analysis with more sophisticated assays like single-cell sequencing or spatial sequencing. And for that reason, Empiri is a 10x Genomics certified service provider. And we, independent of e-slice assays, we provide single-cell sequencing services with every chemistry or platform that 10x offers. But we can also do spatial sequencing as well as single-cell sequencing from e-slices that have been exposed to different treatments from the same patient. So you can make a very controlled comparisons. Just to illustrate what I mentioned earlier, that we can maintain the tissues intact. These are H&E slides from breast cancer, squamous lung cancer, and renal cancer that have been cultured in our system for 8 to 12 days, as you can see. And you can see that the both cancerous portions as well as stromal component is very well maintained in this system. And when we take slices that have been exposed to cytotoxic drugs and just to H&E staining, you can see on the bottom is a tissue that had been exposed. These are control PDX models, same PDX tissues that have been exposed to a cytotoxic drug, and you can see massive cell death on the bottom, and this matches with the viability readouts. So you can imagine all kinds of downstream analysis you might want to do, whether it's multiplex IHC, immunofluorescence or simple IHCs, validating your own targets, and how those targets expression may have changed or cells may changed after testing your agent ex vivo. These results show that E-slice assay is highly reproducible and selective. In this case, we took metastatic colorectal cancer PDX models and tested them with standard of care chemotherapy. So FOLFOX, FOLFIRI, or FOLFOXIRI. On top is a non-responder model where we took PDX models from 2 different mice on 2 different days and performed E-slice assays under identical conditions. And you can see that the results are highly reproducible. On the bottom is an experiment done with a responder to FOLFOXIRI, and as you can see that, again, on 2 different days, on 2 different mice models, mice PDXs, that we can get the same results. We currently have an NIH-funded R01 project where we're doing similar kind of study with triple-negative breast cancer PDX models. And in this case, we've done up to 4 different tumors on 4 different days and get exactly the same result. Importantly, we have done 2 retrospective clinical studies in collaboration with Dr. Scott Kopetz at MD Anderson to see how accurately we can predict patient responses to targeted therapies and chemotherapies in metastatic colorectal cancer. So Dr. Kopetz performs many clinical trials, and in these 2 different clinical trials, he also generated PDX models from the patients who are enrolled in the study. As you can see, in the middle is a patient response to the care during the combination during clinical trial. Blinded to the patient response, we just received these PDX models from corresponding patients, performed E-slice evaluation against dabrafenib and trametinib, and we matched the results after we finished our analysis. And in the second study, the combination was irinotecan plus cetuximab, plus or minus vemurafenib, and same kind of study design. And what you can see is that our ability to match patient response is 100% when we consider standard stable disease and progressive disease as non-responders, and partial response and complete response as responders. So under those criteria, our positive and negative predictive value is 100%. But how much of the tumor microenvironment can we really maintain? So in this first experiment, we used a mouse MC38 colorectal cancer that has a lot of T and NK and cell infiltration into the tumor microenvironment, and performed single cell sequencing by dissociating the primary tumor with no culture versus 8 days after culture in our system. And this normalized bar graph shows that all cell types are present even after 8 days, including NK cells in pink and T cells in red. And when we take the T cells and do de novo clustering and analysis to see whether we can maintain the different T cell activation states ex vivo, we can see that indeed we can. So the cells that are important for a lot of immunotherapy targets, T cell targeting, immune checkpoint targeting agents, CD8 T cells in exhausted and precursor exhaust states are maintained, and we may get some maturation of the early activated cells in culture in 8 days, but we cannot be 100% certain since these tissues are coming from different regions of the mouse tumor. We've performed a similar experiment with human tumors. In this case, is a GBM patient tumor. GBM is very different in that it's a very immune cold tumor, and there are very few to little T cells that infiltrate the patient tumor. And in this particular patient, there were no T cells actually that we could recover, but there were a lot of myeloid cells, which is pretty typical for GBM patients. So UMAP in the middle by assignment will show you that myeloid cells are in dark blue, glioma cells or tumor cells are in orange, endothelial cells in green, and oligodendrocytes, which are brain glial cells, are in purple. So if you actually look at the real numbers of cells indicated in different colors within each bar graph by cell type, you can see that the myeloid cells are maintained in our culture system up to 8 days. And whether you look at the absolute number or proportions of number, those cells are very well maintained. And I showed you in the beginning of the talk that there are 9 different molecular subtypes of myeloid cells in GBM patients. And we wanted to understand whether these myeloid cell fates change or we can maintain those exactly same cell states or phenotypes in our ex vivo system. So again, using the gene signature for these 9 different myeloid subtypes, we asked what subtypes are present in this particular patient and whether those subtypes are maintained over cup time. So when we do, again, de novo clustering of myeloid cells only from this patient tumor, we determine that the majority of the cells, 90% of the cells, are suppressive macrophage 1. And there's a small portion of MDSCs and these MHC positive activated microglia. And as you can see from the bar graph on the right, that the proportions of cells stay more or less the same even after 8 days. And the dominant population in this patient, even after 8 days of culture, is again suppressive macrophage 1. So these results show that not only do we maintain the number of tumor-associated immune cells ex vivo, but also we maintain their native activation states even after 8 days of culture. And we've done similar experiments now with additional cancer types from patients, lung and head and neck cancer, for example. And each paired bar graph showed baseline versus 8 days of ex vivo. And you can see that even fibroblasts, as well as NK cells, monocytes, and CD8 T cells and Tregs are all maintained in our system. So we wanted to know whether we can really measure immunotherapy responses, checkpoint inhibitors specifically, using our system. So we have an NIH-R01 collaboration between my lab and the company and MD Anderson. And we decided to test in triple-negative breast cancer patients, where now the standard of care includes pembrolizumab, Keytruda, plus neoadjuvant chemotherapy. And the clinical trial that led to the approval of this combination, called Keynote 522, they observed that the actual percent of patients who benefit from having Keytruda or Pembro is 13.6%. So about 40% of the patients respond to the neoadjuvant chemotherapy anyway. And if you add Pembro, another 13.6% show this pathological complete response at surgery. And about another 40% of the patients are resistant to the combination therapy. But what's noticeable is that the time course for determining a patient response in the clinic actually is about 24 weeks. Whereas in our ex vivo system, it's 8 to 12 days. So there's huge difference, and this can be used in real time to determine whether a patient is really responding to this very expensive and potentially toxic therapy. So as you know, Pembro costs about $150,000 per patient per year, and the estimated average cost per patient is about $1 million. But it's only about 12 to 13% of the patients who will really respond to this added treatment. So we wanted to see whether we can identify patients who would respond to Pembro ex vivo. So we are taking biopsies from patients who are coming into MD Anderson for receiving the combination treatment, and then measuring responses ex vivo using e-slices. And what you can see is that thus far, our results show that 2 of 7 patients respond to Pembro. And if we make the cutoff to a complete response, only 1 of 7 patients may respond. Actually, it's now 1/9, which is almost exactly the kind of percentage they see in the clinic. And we see about 58% patients who respond to the combination, meaning that this ex vivo system can maybe very faithfully recapitulate clinical responses, at least for triple-negative breast cancer patients in the clinic. This is an ongoing study, and we will be analyzing another 25 or so patients, and we're very excited about the preliminary results we have seen so far and looking forward to the completion of the study. And we will match our responses ex vivo to the patients. But in an earlier study, we also looked at whether we can measure nivo response in lung cancer patients. And this was a smaller study and with less funding. So we didn't get to match patient response to the ex vivo response. But what we saw was only 2 out of 8 patients responded to treatment, which is, again, sort of the rates you see in the clinic. And importantly, in this case, we measured interferon gamma levels in the conditioned media as a readout whether the nivo had a positive effect in activating T cells again. And as you can see, so from the same patient, only the wells that contained nivo treatment showed increased interferon gamma response, consistent with the viability changes we measured. So, and we only observed this only in responders, but not in non-responders. So biomarkers, as well as just the proportions of patients showing response in our ex vivo system, matches what is expected in the clinic and as a biomarker. So you can imagine this platform is highly flexible, can be used in many different contexts in clinical and preclinical settings. So in the clinical trial, for example, it can be used to do patient selection upfront. It just as 7 days after biopsy to determine whether a patient will likely to respond or not likely to respond to the new regime. And of course, preclinically, you can use this to identify the most promising indication for your new drug before you commit to a clinical trial. It can be used to testing drug sensitivity and efficacy, mechanism of action, on target effects. It can be used to test drug combinations and sequencing. And I think also very importantly, and I think it's a beautiful way to really discover biomarkers in a sort of unbiased screen in a highly efficient manner. And I will show you some data, maybe I don't, but that it can be used for mechanism resistance and drug screening as well. So this was from a prospective correlative study from an ongoing clinical trial with the combination being tested was E plus N. And we knew that there is a potential for resistance to the target therapy combination E plus C. That can be inhibited by an additional drug called B. And what you can see is that different color bars represent different days of ex vivo culture. This is all from the same patient from an 18-gauge biopsy core, that the combination of E plus C or E plus N suppressed or controlled tumor growth ex vivo up to day 12, which is the gray bar, or on day 16, which is the yellow bar, and day 20 on blue bar, there was increased in viability, suggesting that there may be a resistant clone that emerged and outgrew in the culture. However, in the green outline boxes with arrows following the same condition, where we added additional inhibitor B, we can suppress this emerging clones at day 16 and day 20. So in this particular study, we knew what the potential mechanism of resistance may be. However, if you're not aware or you want to discover novel mechanisms of resistance, you can imagine you can take the samples pre and post emergence of resistance clones and perform molecular analysis to identify what those mechanisms may be in human tumors. I think that's really important, that this is not a cell line, this is not a mouse model, this is not, ex vivo manipulated systems. So it's a patient tumor with an intact ecosystem and tumor microenvironment. We also performed in my academic lab drug screenings using 2 GBM patient samples. So this platform is really not ideal for doing massive drug screening with 100,000 compounds, right? I think maybe some of the other platforms may be better suited for that. However, if you you have narrowed down your potential targets or compounds to, let's say, tens or a couple hundred compounds, we can perform in patient tissues, fresh patient tissues, these drug screening. And in this case, we identified 5 drug candidates or drugs from a library of 484 CNS penetrant drugs that overlapped between patient 1 and patient 2. Then we took one of those compounds and tested an additional independent GBM patient tumors and observed nanomolar range activity, suggesting that yes, we could use this as a pre-screen before you go into the clinic to narrow down your promising leads. So just to summarize, comparing e-slices to organoids to 3D cultures and 2D cultures, TE slices offer many advantages, including and most importantly, the maintenance of the native 3D tumor environment. And what's not listed here, but probably was obvious from the previous slides, is that we can measure treatment responses in 4 days for chemotherapy or cytotoxic drugs, but we can also maintain these cultures out to 30, 40 days in case you want to study resistance mechanisms. And it can maintain specific patient-specific extracellular matrix and the tumor microenvironment ex vivo, and it does not contain serum or artificial factors or chemokines and cytokines that can stimulate or change the immune or cancer cell behavior ex vivo. And because of our internal controls and initial quality controls, we can account for spatial heterogeneity and cellularity by measuring absolute response from each tumor tissue. And it maintains the tissue architecture after even 8 to 12 days in culture that allows histological as well as spatial sequencing analysis, for example, to really delve deep into what the mechanism of action of your agents or drugs is in different cell types. Oops, sorry. I thought that was a little bit out of slides. Anyway, I would like to just end here and highlight and compare our ex vivo system to other systems that are out there for predicting patient responses. And they're either histology-based, genomic sequencing-based, or 3D cultures like organoids and organoid chips or PDX. But in terms of faithfulness to the patient tumor, e-slice is the closest because there is no additional manipulation, and we expose to the drugs right after we take the samples out of the patients. And in terms of cost, it's also quite manageable and affordable. And with that, I can stop the presentation and take any questions you might have.
Houston, we do have a handful of questions here.
Okay.
Let's see. First one is, you mentioned examples of small molecule therapeutics and antibodies. Is the platform amenable to cell-based therapies?
Yes. So we have done studies with NKTs and other NK cell modulating studies, but we can also do it with CAR-Ts, obviously.
Okay. And then you mentioned defined media. What about hormone-dependent tumors?
Yeah, that's a great question. So our base media does not contain any factors, but we definitely can add hormones or cytokines depending on your experimental goal. And so for ER positive, PR positive breast cancer, we've just done an experiment study with that. We do add estradiol.
And a follow-on question to that, proteomic and metabolomic analysis of either the tissue or the culture media.
Yeah, so we haven't done a metabolomic study, but as part of our R01 study, we are doing proteomics analysis from the conditioned media and eventually with the tissue as well. So the conditioned media, I have used them to do mass spec analysis as well as O-link or cytokine arrays. So it can be used in any platform, depending on the sensitivity assay and the abundance of the molecule you want to detect. But it has been tested in mass spec settings as well.
Okay, and then the last question, the 10x services, does that include spatial profiling?
Yes, yes, it does. In fact, we do Visium as well as Visium HD. We have a lot of experience and can produce beautiful data for those.
Okay, that wraps up the questions. For all of the participants, you will be receiving an email follow-up that will have my contact information. If there are further questions, we're happy to answer those by email. And we are, of course, happy to speak with you about pilot studies or accessing the platform as needed. So thank you very much. On behalf of Empiri, I very much appreciate your participation and hope to hear from you all very soon. Thank you.
Thank you.